talking about optimizing a technique that is going to be used to quantify a very diverse group of viruses that until recently have been kind of overlooked in the marine environment. And this is mainly due to methodological limitations. Before I get into that though, let's go back and review why viruses are important. So as I'm sure you heard in Natalia's talk earlier this morning, viruses are the most abundant biological entities on Earth. And even though they are really small, they still make up the second largest biomass in the oceans following bacteria. Because of their high diversity and their high abundance, it's pretty much assumed that all organisms have a virus that infects it. Since the most abundant living organism are bacteria, most of these infect bacteria, and we call those bacteriophage, or just phage for short. So as bacteria are really important in nutrient cycling in the ocean, so are the viruses that infect them. You can think of most of the times a viral infection of a bacteria results in the cell death or breaking open of that bacterium, releasing nutrients back into the environment. So how do we study these things? Normally, to quantify their abundance, we can stain their genetic material. So this is a cyber gold stain, and we put it under an epifluorescence microscope, and we get this beautiful picture. These large circles right here are bacteria. They have more genetic information, so they appear to be much larger. And then the small ones are viruses. Another way to study viruses is to look at their diversity, and a lot of times we do this with metagenomics. And that's basically taking all of the DNA in a sample and sequencing and understanding what the viral community looks like. Unfortunately, until recently, both of these methods overlooked a pretty prevalent group of viruses known as the gokushoviruses. So gokushoviruses have a very small genome, so small that when we stain it with cyber gold, they don't produce enough fluorescence to be able to see under a microscope. And in the case of metagenomics really is biased towards double-stranded DNA. Now, the gokushoviruses only have one strand of DNA, so they were overlooked then too. So how do we know about them? Well, in 2006, a new technique known as rolling circle amplification came along. And what rolling circle amplification did was it preferentially amplified small circular genomes. And guess who had a small circular genome? That's right, gokushoviruses. So since we started adding RCA before metagenomic studies, we started finding them everywhere, in animal and human guts, in freshwater systems, marine systems, and sediments, just to name a few. But because the techniques we use either bias for or against gokushoviruses, we have no idea about how abundant they are. We know they're very diverse, we find them everywhere, but what does that actually mean in the big picture? So how do I plan to quantify them? Well, I'm using a modification of a very classic molecular technique known as the polymerase chain reaction, or PCR. So as a reminder, what PCR is mainly doing is it's just making a lot of copies of a section of the gene we're really interested in. So if we have this template DNA up here, we use two molecular markers known as our primers that are going to attach to a highly conserved region on that gene. They are then going to amplify that center region and make many, many copies. So after multiple cycles, we end up with over 1 billion copies of what we started with. And that's one way that we can increase our DNA so we can either sequence it or even visualize it. Though there are quantitative techniques of PCR, they only work if you have a highly specific gene that's conserved in what you're looking for. Unfortunately for viruses, that's really hard to find. Even closely related species are highly divergent from one another. So instead of using quantitative PCR, I'm using a solid phase PCR known as the Polony method. And so what that means is that I'm basically trapping viral particles in a gel so that they can't move, and then carrying out PCR in that spot. Now I'm creating enough DNA sequences that I can attach a fluorescent probe and visualize it. Then I count all of those probes, all of those PCR colonies or Polonies, and I can extrapolate the quantity of viruses from there. So I'm going to walk through this a little bit step by step. So just to give you an idea on how small of a scale this is happening, up there is the microscope slide that this all takes place on. That oval right there is where the gel is. I've made the gel a lot bigger here, and you can see in the gel are your viral particles in yellow and this anchored primer in red. So the anchored primer is always going to stay stuck onto the gel no matter what. Even though the anchored primer is present throughout the gel, to make this easier for visualization, we're only going to focus on a few spots. So the first step is to heat up everything, and that causes the virus to break open and it releases its genetic information. Now the primer can actually bind and replicate the genome from there. You go through this cycle again, you heat it up, causes the strands to separate, and then we can have that replication. In addition to that anchored primer, I also have an unanchored primer, again, throughout the whole gel that carries out replication in the other direction. That just is going to help us make a lot more strands a lot faster. So this carries out a few more times, and eventually when you wash away anything that's not attached to the gel, you will get these strands of anchored primer that are stuck onto that gel, and only in those areas where we had viral particles in the first place. Those are our Polonies or PCR colonies. Now we can use a fluorescent probe that attaches to an internal region of this strand that fluoresces for visualization. So we visualize these using a microarray, and the result is that picture over there. Each of those dots represents a Polony and thus represents where a viral capsid once was. So we're doing this work with our collaborators in Israel in the Glendale lab. They're the ones that optimize this technique for viral quantification, and they became really interested in what the gokushovirus diversity was in the Red Sea, and so that is where all of my samples actually come from. So just to orient yourself, the Red Sea is between Egypt and Saudi Arabia, and we're actually collecting samples in this offshoot, the Gulf of Aqaba, at the most northern point, because that's where it actually intersects with the southernmost part of Israel. The Gulf of Aqaba actually becomes fairly deep fairly quickly, so we can sample from about 0 to 400 meters. I needed to get an idea of what the gokushovirus diversity was before I designed my primers and probes to optimize this technique for them. So we collected some water, put it through a 0.2 micron filter. Only viruses can pretty much pass through this, so I collected the flow through. And then I extracted that DNA, sent it off for sequencing, and resulted with this. So this is just a screenshot of kind of the sequences we get back. And what I'm looking for is for those highlighted areas. That means that part of the sequence was conserved across all of my sequences. You can also see the little stars on top. So even though this region isn't completely conserved for viruses, this is really good enough, and that's where I designed my probe. I looked for a similar conserved region and designed my primer, another primer off of that as well. So here's just a glimpse into the gokushovirus diversity from the September samples that I designed my primers and probes off of. This is just a phylogenetic tree showing you the relationship between the different sequences, and each of these branches show a specific sequence. The dots represent an isolate, and each of the colors represent what depth that isolate was recovered from. So you can see that some sequences were found in many isolates across multiple depths, while other sequences were found exclusively just at that one depth, and in the case of 400 meters, there was no other sequence found at 400 meters. Now I needed to pick my positive controls that worked best with my primer and probe, so I picked 5 that were kind of divergent for each other, because again, we're looking for the quantifying the abundance of kind of a diverse group, and so I picked these 5 here. And so then I was really excited to finally try Polonies on the positive control, which has never been done for gokushoviruses before, hoping that at least one of these positive controls will work. And I got really excited because all 5 of them did. But I celebrated maybe a little bit too soon because there was still a lot of work to be done. So we're really aiming for an efficiency of the Polonies, meaning how many Polonies I know I'm putting into my gel versus how many I'm actually calculating. I want that efficiency to be between 20 to 50%. Unfortunately, I was getting between 8 to 24%. So there was a lot of more hurdles that needed some jumping. I needed to increase efficiency, remove the contamination, and also make sure that when I dilute things, I can when I dilute things, I'll be able to get that consistent efficiency. So I could increase the efficiency. That was just tweaking the PCR protocol a little bit. I removed my reagents and started anew, and I was able to remove contamination. But where I keep getting stuck is this finding that consistent efficiency between dilutions. So what I'm talking about here is that in Israel, this is what they do with the viruses that they work with. So each of the different colors just represent a different viral group, while the x-axis is showing you the number of viruses they know they're putting into their sample, and the y-axis shows the number of viruses they're counting. This should be a nice linear relationship if we want to use this as a quantitative technique to be able to back calculate to what we're seeing in the environment. So as you can see, their lines get nice R-squared values of 0.97 or higher. When I do the same exact thing, I'm getting under 0.8 at best R-squared value. So that's where I'm kind of stuck at right now and have to be able to optimize first before I will be able to actually quantify this in the field. So just kind of as like a summary, so we do have positive controls working with Polonies, but right now I'm really working on tweaking those primers and probes so I can get those to be more consistent efficiencies each time. This is really important, again, for being able to go back to the field samples. Also, we did see a lot of diversity in September, which was really interesting. I have samples across 3 years, 4 different seasons at all of these depths. So along with their abundance, I'll also be looking to see how their diversity changes as the water column changes in the Red Sea. So with that, I'd just like to say thank you to a lot of people and open it up for questions.